Hello and welcome everyone to the CarboHydrate Chronicles, a podcast about glycoscience and glycoscientists. In this show, via a series of interviews, we explore the world of carbohydrate science and innovation from a personal point of view. Your host today is Tamas Shahai, a member of CarboHydrate's scientific team. Developing drug delivery systems is a challenging area of the pharmaceutical industry that face real challenges in the 21st century, like the need for targeted delivery, or triggered release, personalized therapy, effective formulation of gene and cell therapy drugs, and the list goes on endlessly. Cyclodextrins in the classical sense have limitations in these fields as they cannot target tissues or release their cargo at will, unless we apply some tricks to them and chemically modify them so that they can fit such novel applications. Today, we have with us Professor Francesco Trotta, professor at University of Torino, vice president of the European Cyclodextrin Society, who can tell us more about these challenges as he's a pioneer in advanced drug delivery systems and nanotechnology. Francesco will introduce us into the nanoworld of cyclodextrins with all its possibilities and future prospects. Welcome, Francesco. Thank you for accepting our invitation.
You are welcome. Thank you so much for giving me this opportunity. And I am very proud to speak about my career regarding the cyclodextrin field. I fall in love many decades ago and still continue.
By way of a bit professional introduction, Professor Trotta's main research relates not only to cyclodextrins, but pharmaceutical technology in general. He authored over 100 papers, several key reviews and books on drug delivery, along with numerous patents. He is a member of over a dozen Italian international associations, including both the European and the global cyclodextrin societies. Francesco, when checking your life journey, we can see that you've been working at the University of Torino for over 32 years. Are you originally from Turin?
No, I was born in a very small village in Cilento region in the south of Italy 64 years ago. Now this region is a World Heritage by UNESCO, but at that time it was a very poor region. Many people tried to move from the south of Italy to other countries in Europe, also in the world, or in the richest north of Italy. One of those people was my father. He decided to move the family to Asti. Asti is a small town close to Torino.
And famous for the champagne.
Yeah, a very good wine in not only for, but good wine was in Asti region, and attend all the school up to the college.
Do you often visit the region and the village you were born?
Yeah, sure. I have also a house there that was of my grandfather, and during the summer I always go there. I also I like to grow olive tree for olive extra virgin oil production for family use. And I like so. In addition, last International Summer School on cyclodextrin was held in Ascea, and Ascea is very close to my village.
Why have you chosen science as a profession? Does it run in your family?
No, absolutely no. My father was just work in Fiat car factory in Torino. But I like science from when I was from the middle school. No, all the science like me, in particular chemist. What happened when I was very young and a student in the middle school in Asti? With small saving, I collect my money and I bought two games that at the time was very popular, The Little Chemist. And in the afternoon, I spent a lot of afternoons in carry out the experiment described on this in this game. But after a short time, they don't satisfy no longer me. And of course, I try to organize, to design my personal experiment, but the basis was not so strong, and you can imagine what the number of breakage of the glassware, some flames, and try to imagine what is the behavior of my mother and the punishment that I received at the time. But this was, I fall in love with chemistry, and it is quite natural to enroll at the University of Turin. It is famous in the world, even because it was the university where was graduated Amedeo Avogadro.
This is beautiful. I cannot really imagine the little Francesco doing chemical experiments and showing
but the incredible things that there is a small part of that game that was saved after a long time, and still I store in my home now. I have small memory of these games still in my home.
Your road to science and chemistry came from the early ages. When did you first meet or interacted with cyclodextrins? Do you remember that?
At that time, in Italy, it was not a bachelor, but to get the master degree, you have to spend at least 1 year in the lab for experimental thesis. And one of the teacher proposed me to test cyclodextrin, but at that time was the first thesis in my university for sure on cyclodextrin, because I chose cyclodextrin because it is a very new topics at the time for me, and some curiosity and recklessness also because I was young. Thesis was on the modification of this cyclodextrin to use as an alternative catalyst to enzyme. No, after I have stopped because after the master degree I moved to organic chemistry, and I got the PhD working on phase transfer catalysis, a typical organic chemistry topics, but this was, in any case, good for me in the following because it gave me the basis for manage the molecules, to modify the molecules as happened also for cyclodextrin. When I won the position for assistant professor at the University of Torino, I came again to study cyclodextrin step by step, not only cyclodextrin at the beginning. I was also interested to produce some polymer at the time, linear and soluble polymer. Cyclodextrin has a lot of functional groups that can react, and to synthesize only linear and soluble polymer is quite a hard task. But after that, I was in the literature, I saw a paper of researchers from Los Alamos, Li Quan Ma, that are working on the cross-linking of cyclodextrin, producing a very impressive result with in the bioremediation, producing the so-called nanosponges. Mm-hmm. I was fascinated, but I realized that I have in my hand many kind of nanosponges. I try to focus on this synthesis and to find a lot of different applications, not only for bioremediation, but for drug delivery, to a large extent, in energy application, in CO2 captures, and so on. So actually, what was for a byproduct in the first attempt in the synthesis of polymer cyclodextrin was my best results, my achievement of my career.
So you can never know if a failure is actually a failure or a success. You just need to find out the way you use it.
No. So, actually, forget to remember that in my lab, my students, when I discuss with them, I say that we follow the pig chemistry. Why pig chemistry? You know that the pig in Italy, but I suppose also in other countries, never is waste. All the part of the pigs has some application use. There is nothing that we have to discharge by using pigs. The same in our lab. We are not so great to waste some of our experiment. We have always to follow an application, a use of our experiment, our material, and so on.
That's a very nice philosophy. I really like it. Pig chemistry. That's
Exactly.
What is the root of fascination? What do you enjoy most about being a scientist?
As I said before, I am fascinated by all the science, probably, and in particular chemistry. But in the world of the molecule that we found in the natures, carbohydrate is in all the part, even in DNA, there is some sugar, let's say.
Yes.
So probably is the brick that is present in every stage of our life of the world. So it's for me particularly fascinating. And even because there is only 3 atoms, carbon, hydrogen, and oxygen. But with this only 3 atoms, you can get a lot of incredible number of molecules, application, use, particularly fascinating.
Yeah, it's an unlimited source of challenges for sure. I also realized that you have a lot of granted patents. You are scientific advisors for many Italian companies. With CarboHydrate, we also have a collaboration. Now, one of your students is probably doing hard work in the lab as we speak.
Yeah.
How come that did you ever consider going for an industrial career in the private sector or academia is your field?
The pathway of my career is quite changed because when I was a student at the university, there were 2 possibilities to get the master degree in industrial chemistry or in the pure chemistry. I follow the pure chemistry because I hate industrial application, money. So I prefer to imagine ideal chemistry. No? Now I am a teacher of a full professor of industrial chemistry. But I realized that during the time, many years, that the chemistry can help humankind if it is very simple. So industrial application generally require very few modification, no organic solvents. If you make a very simple reaction, you can arrive to a product that is possible to scale up, but this means that you can diffuse the application in a large part of the people. And fortunately, nanosponges is generally one step, very high yields, sound good to industrial application.
When you do your research, you also consider that eventually, if it's good and the discovery is promising, it should be ready to be taken to the next level, not just for the pure research itself.
Research should don't have at the beginning any prerequisite. You have to investigate because you believe that topics that research is of interest for you and probably for many other. But after that, you have also to consider if this research can move from academic role, academic field to industrial field. If you see, for instance, in the field of cyclodextrin, there is many use in bioremediation. A very important teams produce some cross-linked cyclodextrin actually by using nanosponges, and with very good results in removing some pollutants. The problem that the synthesis was performed in DMF by using toxic cross-link and so on. So this is a very interesting for a publication, for a paper, but the application has no sense. And I don't like if our work has no possible application, at least.
I see. We've been talking a lot about your education activities and your research activities. Do you prefer either teaching or lab work, or you like the balance?
Okay, because I am a professor, teaching is my role. But if I have to make a selection, I prefer research that teaching. But after that, it's not so different, because if you have in your hand a good research, new results to share, this is also positive for teaching. Otherwise, you always repeat the same sentences year by year. And if you have some research results, you can share new findings with your students.
It's also easier to inspire people, motivate them, show them that research can have a meaning and purpose.
Right. To pass to the student not only the lesson, let's say, all the topics of science and so, but also the motivation, the enthusiasm for research. Otherwise, it is not only the translation of some part of information.
When you're not in the lab, what do you do in your free time? You have any hobbies?
Okay. First, when I have free time. My preferred job is to be grandfather. I have two. I have a grandson, Matteo, and granddaughter, Melissa. I regret to have not so time to spend with them, but I like so much. The second is that even before that I became a chemist, I was a student of karate. And I am also a master of karate, and 2 times a week I have to follow my karate team, and I like so much. And the third, probably, I like gardening. I like to grow flowers and the small vegetable in my home. That actually is not Turin, not Asti, but is in Genoa, where is my wife. And so I have 3 towns I have to cross every day. Asti, where is my daughter and my nephew, Turin, my job, and Genova, where is my wife. So there is a quite difficult town to manage.
It sounds like a busy life, yeah. But I'll keep that in mind, that karate part, and mind my tongue when we meet next time. What I'm saying, I will be more careful about that. Let's talk a little bit more about the research that you're doing. Thinking about a global picture or a bigger picture in nanotechnology or drug delivery, what are current trends you are most excited about?
To my opinion, we have to follow and to spend much time in targeting delivery. To my opinion, cyclodextrin and cyclodextrin derivatives, polymer, and so on, can have a great benefit to make much precise medicine. With the selective modification of cyclodextrin, we can drive the drug to selected organ and cell. There is a lot of work that was already done, but probably there is even much more to do. The second is the gene delivery, and I think that there is a lot of work to do, and it is also one of my topics in this year. We also have financial support from quite large support, financial support from our ministry. And again, I would like to stress much more the audience, the nutraceuticals delivery by cyclodextrin or cyclodextrin derivatives, because actually nutraceuticals is a big challenge in the science. There is a lot of formulation that is selling even in internet, or very small efficacy. Probably we have to study much better this stabilization, solubilization by using cyclodextrin or cyclodextrin derivatives. This because, in particular, I have some interest on the central nervous system disorder, say Parkinson's disease, Alzheimer's disease, ALS, and also Huntington disease, that many of these nutraceuticals that we can find in the nature can have a great benefit. Probably not curing completely, but try to help to improve the quality of life. To my opinion, we can obtain some benefit if we were able to, for sure, to follow some diet, for instance, that can help in general the wellness or the health of the people. Mm-hmm. Even because I was born in the region where the Mediterranean diet was found, I try to follow this kind of diet. But there is many nutraceutical, many molecules from natural origin that can have a great effect. So gene delivery, target delivery, and nutraceutical, maybe in the case of medicine, is the topics that for me that you should have to study.
These are indeed very fashionable topics, and a lot of research are focusing on these indications in the 21st century. If you look back at your career and all the research and discoveries you have made, is there any specific project or accomplishment that you're particularly proud of?
Sometimes if you did a good thing or not is not decided by yourself, but by the other, no? Now we have the citation, the H-index, the publication. If you take a look on Scopus, for sure, I have to say nanosponges, because many people, many colleagues in the world work on these topics that at that time, 20 years ago or 25 years ago, the first paper on these topics for me was just a synthesis. I cannot imagine how this material can found so large number of applications, not only in my group, but abroad.
There are certainly a lot of remarkable research that you have done. And as you said, the applications are partially for the future to discover what they can be used and what are the most practical uses. Talking about the future, with a lifetime of experience, what would you advise for young scientists who are just starting their career in science?
Actually, this means that I am quite old to suggest to young researcher some behavior, some advice. The first is for sure study hard. We are not to limit only to few information on internet, verify all the information that you find, but you have to study hard. The second probably is common for the scientist. Be open mind. You have not to focus only on your idea, to be able to interact and to discuss with other. But at the same time, you have to probably to follow your intuition. If you want to make a great achievement, you have to follow some intuition, some crazy idea sometimes, no? Because in my career, most part of the experiment failed. You have not to never get discouraged if the experiment don't work as you expected. But try to be confident. Failures in my life, not only in the lab, but in the life, was much more frequent than the success, but never discouraged.
But I think that's natural. I mean, failure is not the end of the story. It's part of the learning process. So
it's the beginning. Yeah, right. So if I can produce the negative results that I got in these 40 years of research, I was one of the magnitude much more paper.
Maybe your citation index would not be as high.
For this, we have to follow the peak chemistry.
I just have one last question that all guests have in common. If you could have dinner with a historical figure or celebrity or fictional character that are alive, who would it be and why?
For me, it is easy to answer because I have in mind Emil Fischer. Emil Fischer was Nobel Prize winner in 1902, but because it's very fascinating for me, even just when I was a student, because found a lot of molecules of isomer of the glucose chemistry that was impressive. Not all because at that time we are not NMR, we are not HPLC, we are not instrumentation. He was able to select, to recognize a lot of molecules that very similar each other. No, at the same time he was also the chemist that was able to explain in an easy way the behavior of the enzyme, the lock and key. But if you have to limit to cyclodextrin, I cannot not mention Professor Joseph Saitri that I was proud to know many times, many years ago.
I hope you enjoyed the most recent episode of CarboHydrate Chronicles today, delivered to you by CarboHydrate. We talked with Francesco Trotta, professor from the University of Turin, an expert in drug delivery with a specialization to cyclodextrin nanoparticles. For me, Francesco's message is that you can never know if a failure is indeed a failure, and you should remember the big chemistry, that all parts of the pig has some use and all parts of your results has some use probably. So you just need to find it, study hard, and follow your intuition. Thank you, Francesco, for being with us today. This was a lovely discussion, and I hope to see you soon in person in a few months, probably.
You're welcome. Ciao, Thomas.
Thank you, everyone, for listening for today's interview or discussion. Next week in CarboHydrate Chronicles, we'll talk with Luigi Lei, professor of chemistry at University of Milan and expert in advanced glyconanomedicine and carbohydrate-based vaccines. Your host was Tamas Shahida from CarboHydrate. I hope you'll join me in our next episode too. See you in 1 week. Goodbye.